and welcome dear students. Today we are going to discuss the key topic, that is phagocytosis, pinocytosis and exocytosis. The main objectives of today's lecture are to have a detailed idea about the process and mechanism of phagocytosis, to discuss in detail the process of pinocytosis, to get an idea and explanation of cell-mediated endocytosis, to discuss the process and mechanism of exocytosis, to have a general idea about the role of immune system in phagocytosis, to know the difference between phagocytosis and pinocytosis. Dear students, let us first of all discuss about phagocytosis. Phagocytosis or cell eating, first described by Metchnikoff, is the process by which a cell engulfs a particle and digests it. The word phagocytosis comes from the Greek phago meaning devouring and cyte meaning cell. Cells in the immune systems of organisms use phagocytosis to devour bodily intruders such as bacteria, and they also engulf and get rid of cell debris. Some single-celled organisms like amoebas use phagocytosis in order to eat and acquire nutrients. It is an active and highly regulated process involving cell surface receptors and signaling cascades mediated by Rho family GTPase. Now, let us have an idea about the mechanism of phagocytosis. The important steps involved in the process of phagocytosis are as: first step, the cell that will perform phagocytosis is activated. This can be a phagocyte, which is a cell in the immune system that performs phagocytosis, or an organism such as an amoeba, which behaves in a similar way to phagocytes when it carries out phagocytosis. In case of immune cells, activation occurs when the cells are near bacterial cells or parts of bacterial cells. Receptors on the surface of the cells bind to these molecules and cause the cells to respond. Step 2. In the immune system, chemotaxis may occur. Chemotaxis is the movement of phagocytes towards a concentration of molecules. Immune cells pick up chemical signals and migrate towards invading bacteria or damaged cells. Step 3. The cell attaches to the particle that it will ingest. Attachment is necessary for ingestion to occur. Some bacteria can resist attachment, making it harder for them to be taken into the cell and destroyed. Step 4. The cell ingests the particle and the particle is enclosed in a vesicle called a phagosome. The phagosome transports the particle into the cell. Step 5. A lysosome fuses with a phagosome and the particle is digested. A phagosome fused with a lysosome is called a phagolysosome. Step 6. Cellular waste such as broken down molecules that cell cannot reuse is discharged from the cell by the process of exocytosis. Dear students, let us have a look on the examples of phagocytosis. Phagocytosis are found throughout the human body as white blood cells in the blood. Many different types of white blood cells are phagocytes, including macrophages, neutrophils, dendritic cells and mast cells. White blood cells are known as professional phagocytes because their role in the body is to find and engulf invading bacteria. Non-professional phagocytes include other types of cells like epithelial cells, endothelial cells and fibroblasts. These cells sometimes perform phagocytosis, but it is not their primary function. Amoeba perform phagocytosis in order to consume food particles. Amoeba engulf particles by surrounding them with pseudopods, which are temporary arm-like projections of the cell that are filled with cytoplasm. Ciliates are another type of organisms that use phagocytosis to eat. Let's now discuss pinocytosis. Pinocytosis is the method by which a cell absorbs small particles outside the cell and brings them inside. The word pinocytosis comes from the Greek for cell drinking. During this process, the cell surrounds particles and then pinches off part of its membrane to enclose the particles within the vesicles, which are small spheres of the membrane. This process is usually used for taking in extracellular fluid. Let's now explain the mechanism of pinocytosis. An inducer substance such as a protein binds to a receptor on the cell membrane. Then the cell membrane forms a small open-ended pocket or invagination around the part of the extracellular fluid that is going to be absorbed into the cell. The cell membrane starts to reconnect at the open end of the invagination or pinch off. When the cell membrane has fully pinched off the invagination, a vesicle is formed. This is a small sphere of the cell membrane that surrounds the materials that the cell has just taken in. It prevents the molecules from disrupting the rest of the cell as they are transported. The molecules inside the vesicle are eventually released to be used by other parts of the cell. Dear students, let's have a look on the examples of pinocytosis. Pinocytosis is widely used among the cells of the body, but there are specific situations where pinocytosis plays a major role. Microvilli in the gut use this process to absorb nutrients from the food. Cells in the kidney can use pinocytosis to separate nutrients and fluids from the urine that will be expelled from the body. In addition, human egg cells also use it to absorb nutrients prior to being fertilized. Let's now discuss the types of pinocytosis. Pinocytosis is broken down into macropinocytosis or micropinocytosis based on the size of the vesicle that is formed. Vesicles formed from macropinocytosis are about 1 to 2 micrometer in length, while those formed from micropinocytosis are even smaller, about 0.1 micrometer, and are formed from the tiniest indentations of the cell membrane. Dear students, let's now discuss about the receptor-mediated endocytosis. Receptor-mediated endocytosis provides a means for the selective and efficient uptake of macromolecules that may be present at relatively low concentrations in the extracellular fluid. Cells have receptors for the uptake of many different types of ligands, including hormones, growth factors, enzymes and blood-borne proteins carrying certain nutrients. Substances that enter the cell by means of receptor-mediated endocytosis become bound to receptors that are collected in specialized domains of the plasma membrane known as coated pits. Receptors are concentrated in coated pits at 10 to 20 times their level in the remainder of the plasma membrane. A coated pit invaginates into the cytoplasm and then pinch free of the plasma membrane to form coated vesicles. The macromolecules to be internalized first bind to specific cell receptors. Most of these receptors are concentrated in specialized regions of the plasma membrane called Clathrin coated pits. With assistance from the membrane associated GTP binding protein dynamin, these pits bud off from the membrane to form small Clathrin coated vesicles containing the receptors and their bound macromolecules. The Clathrin coated vesicles then fuse with early endosomes, in which their contents are sorted for transport to lysosomes or recycling to the plasma membrane. The mutations that prevent the LDL receptor from concentrating in coated pits lie within the cytoplasmic tail of the receptor and can be as subtle as the change of a single tyrosine to cysteine. Receptor-mediated endocytosis is a major activity of the plasma membrane of eukaryotic cells. Extracellular fluids are also incorporated into coated vesicles as they bud from the plasma membrane. So, receptor mediated endocytosis results in the non-selective uptake of extracellular fluids and their contents in addition to the internalization of specific macromolecules. Clathrin coated pits typically occupy 1 to 2% of the surface area of the plasma membrane and are estimated to have a lifetime of 1 to 2 minutes. Cells also possess clathrin independent pathways of endocytosis that involves the uptake of molecules in cavuli, small invagination of the plasma membrane that are organized by caveolin. Caveolins are a family of proteins. In addition, large vesicles can mediate the uptake of fluids in a process known as macropinocytosis. Thus, while clathrin dependent endocytosis clearly provides a major pathway for the uptake of specific macromolecules, cells also use several clathrin independent mechanisms. Receptor mediated endocytosis differ from the phagocytosis or pinocytosis in several respects. Receptor mediated endocytosis allows cells to take up specific macromolecules called ligands, such as proteins that bind insulin, transferrin or low density lipoprotein cholesterol carriers. Receptor mediated endocytosis requires specific membrane receptors that recognize a particular ligand and bind to it. Ligand receptor complexes migrate along the membrane surface to structures called coated pits just inside the cytoplasm. These pits are bordered with the protein clathrin, which can polymerize into a cage shaped structure. When such clathrin cages form, they pinch off an enclosed membrane vesicle. Clathrin coated vesicles move into the cytoplasm, selectively carrying the ligand from the extracellular fluid into the cell. Materials bound to the ligand such as iron or cholesterol may be moved into the cell, while the empty ligand returns to the surface or the ligand may be destroyed. Dear students, let us now discuss the differences between pinocytosis and phagocytosis. Pinocytosis and phagocytosis are similar processes, but they have some key differences. Phagocytosis is used to absorb materials much bigger than the particles that can be absorbed by pinocytosis, such as bacteria. Phagocytosis involves the ingestion of solid materials, while pinocytosis involves the ingestion of liquids and solute particles. During pinocytosis, the contents of the vesicles are emptied directly into the cell, but this does not happen in phagocytosis because the contents of the vesicles are too big. Lysosome must combine with the vesicles to break down the contents. This process does not occur in pinocytosis. Let us now explain the process of exocytosis. Exocytosis is the cellular process in which intracellular vesicles in the cytoplasm fuse with the plasma membrane and release or secretes their contents into the extracellular space. Exocytosis can be constitutive or regulated. Constitutive exocytosis is important in transporting proteins like receptors that function in the plasma membrane. Regulated exocytosis is triggered when a cell receives a signal from the outside. Dear students, let us now discuss about the mechanism of exocytosis. The mechanism controlling regulated exocytosis were largely discovered in 1990s. Contrary to early ideas, membranes normally do not fuse together spontaneously. This is due to the negative charges associated with the phospholipids that make up the lipid bilayer of the membranes of vesicles and organelles. Membrane fusion requires energy and the interaction of special adapter molecules present on both the vesicle and plasma membrane. The adapter molecules are highly selective and only allow vesicles to fuse with membranes of particular organelles, thus preventing harm to the cell. Once the appropriate adapter molecules bind to each other, energy stored and released by ATP forms a fusion pore between the vesicle membranes and plasma membrane. The contents of the vesicle are released to exterior of the cell as the fusion pore widens. The vesicle ultimately becomes part of the plasma membrane or is recycled back to the cytoplasm. Exocytosis is also involved in the elimination of waste from the cell. Two types of secretory pathways are involved in exocytosis. First one is constitutive secretory pathway and second one is regulatory secretory pathway. Constitutive secretory pathway: protein molecules in the Golgi apparatus which are not tagged with signal peptides are secreted by the constitutive secretory pathway. The products which are secreted out by the cell are sorted in secretory vesicles. Since these vesicles contain a dense core, they are also called dense core vesicles. Small molecules like histamine and proteins like hormones and digestive enzymes are the products secreted by cells. Proteins which are destined for the secretion are called secretory proteins. Once these secretory proteins are loaded into secretory vesicles, they undergo post-translational modifications like the liberation of active molecules by proteolysis. Regulated secretory pathway: the secretion can be activated by extracellular signals as well. This regulated exocytosis is called regulated secretory pathway. Histamine is secreted by mast cells. Once a soluble stimulant called the ligand is bound to the receptors on the mast cell surface, histamine leads to sneezing and itching, which are accompanied by allergic reactions. Neurotransmitters are also secreted by the regulated secretory pathway. Dear students, let us discuss about the purpose of exocytosis. Many cells in the body use exocytosis to regulate enzymes or other proteins that act in other areas of the body or to release molecules that help cells communicate with one another. For instance, clusters of alpha and beta cells in the islets of Langerhans in the pancreas secrete the hormones glucagon and insulin respectively. These enzymes regulate glucose levels throughout the body. As the level of glucose rises in the blood, the beta cells are stimulated to produce and secrete more insulin by exocytosis. When insulin binds to liver or muscle, it stimulates uptake of glucose by those cells. Exocytosis from other cells in the pancreas also release digestive enzymes into the gut. Cells also communicate with each other more directly through the products that they secrete. For instance, a neuron cell relays an electrical pulse through the use of neurotransmitters. Components of the vesicle and extra neurotransmitter molecules are quickly taken up and recycled by the neuron to form new vesicles that are ready to send another pulse to an adjacent neuron. Neurons need to send many signals each second, which indicates how tight the controls are that regulate exocytosis. Dear students, let us have a look on phagocytosis and the immune system. Phagocytosis is a critical part of the immune system. Several types of cells of the immune system perform phagocytosis, such as neutrophils, macrophages, dendritic cells and B lymphocytes. The act of phagocytizing pathogenic or foreign particles allows cells of the immune system to know what they are fighting against. By knowing the enemy, the cells of the immune system can specifically target similar particles circulating in the body. Another function of phagocytosis in the immune system is to ingest and destroy pathogens like viruses and bacteria and infected cells. By destroying the infected cells, the immune system limits how quickly the infection can spread and multiply. The immune system cells that perform phagocytosis can also use other mechanisms to destroy pathogens inside the phagolysosome, such as oxygen radicals. Oxygen radicals are highly reactive molecules. that react with proteins, lipids and other biological molecules. During physiological stress, the amount of oxygen radicals in a cell can increase dramatically, causing oxidative stress, which can destroy cell structures. Nitric oxide. Nitric oxide is a reactive substance similar to oxygen radicals that reacts with superoxide to create further molecules that damage various biological molecules. Another one is antimicrobial proteins. Antimicrobial proteins are proteins that specifically damage or kill bacteria. Examples of antimicrobial proteins include proteases, which kill various bacteria by destroying essential proteins, and lysozyme, which attacks the cell walls of gram-positive bacteria. Antimicrobial peptides. Antimicrobial peptides are similar to antimicrobial proteins in that they attack and kill bacteria. Some antimicrobial peptides like defensins attack bacterial cell membranes. Another one is binding proteins. Binding proteins are often important players in the innate immune system because they competitively bind to proteins or ions that would have otherwise been beneficial to bacteria or viral replication. Lactoferrin, a binding protein found in mucosal membranes, binds iron ions which are necessary for growth of bacteria. The cell surface receptors used for phagocytosis depend on the type of cell that is doing the phagocytizing. These are the most common ones. First one is opsonin receptors. Opsonin receptors are used to bind bacteria or other particles that have been coated with immunoglobulin G antibodies by the immune system. The immune system coats potential threats in the antibodies so that other cells know it needs to be destroyed. The immune system can also use something called the complement system, which is a group of proteins used to tag the bacteria. Another one is scavenger receptors. Scavenger receptors bind to molecules that are produced by bacteria. Most bacteria and other cellular species produce a matrix of proteins surrounding themselves called an extracellular matrix. This matrix is a perfect way for the immune system to identify foreign species in the body. Because human cells do not produce the same protein matrix. Toll-like receptors. Toll-like receptors named after a similar receptor in fruit flies encoded by the toll gene bind to specific molecules produced by bacteria. Toll-like receptors are a key part of the innate immune system because once bound to a bacterial pathogen, they recognize the specific bacteria and activate the immune response. There are lots of different types of toll-like receptors produced by the body, all which bind different molecules. So, another one is antibodies. Some immune cells make antibodies that can bind to specific antigens. This is a process similar to how toll-like receptors recognize and identify what type of bacteria is infecting the host. Antigens are molecules that act like a pathogen calling card because they help the immune system know what threat it has to fight. With this, we come to the end of today's program. Thank you for watching.